The second part of protein synthesis to make a protein, or it is called translation. In translation, mRNA is translated into protein, and it occurs in cytoplasm or ribosome. The translation is the only process here happens inside the cytoplasm or ribosome. Here we have a cell. It shows up how the information flows from DNA into protein or traits. The DNA is located inside the nucleus, and it is so large, can't pass outside. So we need the information from DNA or the message from the DNA. So the gene is converted into mRNA by transcription. Transcription makes mRNA, and then mRNA is going to leave the nucleus and go to the cytoplasm or ribosome. As you can see here in the cytoplasm, ribosome attached to mRNA, and we have here 5 tRNA landed on mRNA, and they carry amino acids to form protein. This process is called translation. In translation, mRNA is translated into protein by using ribosome and tRNA. How we can read the mRNA? mRNA has nucleotides, and every 3 nucleotides form codon, and a codon gives only 1 amino acid. To make a protein, we need 20 amino acids. There are 20 amino acids, and they have different names. They are 20 amino acids. Here, how we can get amino acids from this chart. We have here codons of mRNA: AUG, GCA, AAA. Each codon gives 1 amino acid. You have to get the amino acid from the chart in front of you. The first codon, AUG. It is called start codon. What's the meaning of a start codon? The translation always starts from AUG. It is the start point or start codon. How we can get its amino acid from the chart? Or how can you read this chart to get amino acids? In this chart has 20 amino acids to make protein. How can you read it? We have in a codon 3 nucleotides. In the chart, you can see the first nucleotide in the left side, second nucleotide up, third nucleotide on the right side. For example, AUG. How can you read AUG and get its amino acid? Please use your finger with me. A, put your finger on the first letter A on the left side and move your finger to the second letter U. Second letter, you get second letter from the up. From up, yes. A, put your finger in A. U, put your finger in front of A and in the U, and then leave your finger here. Just move your eyes into the third letter, right side, G, A, U, G. A, U, G is going to be methionine. Methionine is amino acid. So AUG gives amino acid is called methionine. Let's make another codon. GCA. Put your finger on the G on the left side and move its finger towards C column and leave your finger here. Just move your eyes toward the third letter, GCA. GCA, alanine. Alanine amino acid. Let's do another practice. AAA. I think it is so easy. A, put your finger on A, left side, and move towards A, lysine. It codes for amino acid lysine. You have to write here the names of amino acids beside its codon.
We have here different codons, start codon and stop codon. The translation also starts at start codon, which is AUG. And when the translation reaches stop codon, which are UGA or UAA or UAG, the translation stops and it releases a polypeptide chain, which is protein. How does the translation happen? Or what are the steps of translation? We have to use ribosome, mRNA, and tRNA. Ribosomes have two subunits, large subunit and small subunit. The large subunit has stations, around 3 stations, and small subunit and large subunit bind to mRNA and move through mRNA. While it is moving, tRNA comes and it lands at the station of ribosome. The big or the largest subunit of ribosome has 3 stations where tRNA are landed. tRNA, tRNAs, they come and they have certain anticodons and they carry amino acids and their anticodons are complementary to codons of mRNA. As you can see here, the first tRNA, which carries amino acid or green amino acid, has anticodon UCA, which is complementary to a codon AGU. Codon, it is found on mRNA, and it is a start codon or stop codon. It is a start codon. Here, the translation starts. The tRNA, which carries green amino acid and anticodon UCA, it landed on the codon AGU with its specific amino acid, which is methionine. Methionine. The second tRNA, which carries purple amino acid, has anticodon GCA, and it is complementary to CGU. The third tRNA, which carries purple amino acid, has anticodon UGC, which is complementary to ACG. And then they leave the ribosome, and the ribosome form peptide bond between amino acids to form a chain of amino acids. It is called polypeptide chain. Polypeptide chain means protein. Here in the picture, you can see the big ribosome with two subunits, small and large, and they attach to mRNA, which has codons, and the big or the large subunit of ribosome has three stations where tRNAs are landed, and they carry specific amino acids for specific codons of mRNA. Here, the translation occurs and ribosome attached to mRNA and tRNA lands at mRNA with specific amino acids, and ribosome form peptide bond between amino acids. When ribosome reaches stop codon, the translation stops and polypeptide chain or protein is released. Here in the picture, you can see the DNA found inside the nucleus, and it has your genetic material or your genes. And to take the message or the instructions of these genes, we have to make RNA with blue color. RNA is formed, and then it left the nucleus to be in cytoplasm or ribosome to be translated into polypeptide chain protein. During translation, ribosome comes and attaches to mRNA, and then tRNA comes and lands on ribosome or mRNA with specific amino acids, and ribosome makes peptide bond between amino acids to form peptide chain or protein. Here we have an example of DNA template or gene that have been translated into amino acids. And we know DNA is very, very big, can't leave the nucleus and can't be outside in the cytoplasm to be translated. So we will convert each gene to be mRNA, because mRNA can leave the nucleus and be translated outside in the cytoplasm. And it gives us proteins or traits. DNA here, TACA-TGCGGATT, will be converted into mRNA, and you have to follow base pairing rule. A pairs with U and C pairs with G when you are making mRNA or tRNA, any RNA. T will pair with A. A pairs with T? No, U, because we are making mRNA, RNA. C, G, A, U, because we are making mRNA. T, A, G, C, C, G, G, C, G, C, A, U, T, A, T, A. Now we have mRNA, and it left the nucleus to be translated outside and attach it to the ribosome. And tRNA will come to bind to mRNA, and they bring amino acids. tRNAs bring amino acids, specific amino acids for each codon. What is the anticodon for each codon in the mRNA? We have in mRNA codons AUG, UAC, GCC, UAA. How many codons here? AUG, UAC, GCC, UAA, 4 codons. So they have 4 tRNA anticodons. AUG will pair with UAC. UAC paired with AUG. GCC will pair with CGG. UAA pairs with AUU. tRNAs bring specific amino acids. What are the amino acids for these codons? Which strand here will be read to get amino acids from the codon chart in front of you? mRNA codon or tRNA anticodon? We will read mRNA. mRNA here started with AUG. Perfect. It is a start codon. AUG will give methionine. The first codon, AUG, gives methionine. Second codon, UAC, will give tyrosine. The third codon, GCC, will give alanine. The last codon, UAA, will give stop codon. Okay, here my question. How many amino acids here we had now? 4 or 3? 3, not 4, because the last codon, it gives stop codon, which translation stops, and no more amino acids. We have here 3 codons, codes for 3 amino acids. And the last codon gives stop codon, which stops the translation. Methionine, tyrosine, alanine, they are the amino acids that formed from our mRNA. We have here now 3 amino acids.